welcome to my channel Pure Biology. And today I come with another video that is the continual portion of the translation. And in my previous video I have already explained about the initiation, but elongation is not described in that video. So here the process elongation is explained. Okay, so first, what is the elongation? It is the cyclic process on the ribosome in which the amino acid at a time is added to the nascent peptide chain. So that means elongation means the addition of the amino acid to form the polypeptide chain. Okay, and the peptide sequence is determined by the order of the codons in the mRNA. So how the peptide sequences are determined? The peptide sequence is determined by the order of codons. Okay, how the codons are there in orderly or sequentially, in that way these are determined, and then arrangement or the attachment of the codons with each other to form the peptide chain. And here first, that is the process is known as the decoding. The decoding is the first part of the elongation process. That is, the ribosome selects and binds an amino acid tRNA whose anticodon is complementary to the mRNA codon in the A site. Okay, so what is saying that ribosome, it helps to select and there bind with which one? With the amino acid tRNA whose anticodon is complementary to the mRNA codon. That means it can bind with that amino acid tRNA whose anticodon site must be matched with the mRNA codon. Okay, so without matching of the complementary anticodon, the addition or the attachment is not possible. And here, what is the role of the EFTU? That EFTU, these are the elongation factors, and here the many elongation factors are involved, and I have already explained that elongation factors, so you just check out the previous video where the elongation factors are explained. So now here EFTU, that mediates the entry of amino acid tRNAs into the A site. So where the EFTU, what is the role of the EFTU? EFTU that helps to enter of amino acid tRNA where? To the A site. Okay, there are three sites: A sites, B sites and E sites. So first, where they enter? In the A site. Which one is entered? The amino acid tRNA. Okay, the second point is that first, EFTU that binds to the GTP. Second, the activated EFTU GTP complex binds to tRNA. So first one is that EFTU that binds to GTP, and then when the EFTU form the complex with GTP, this complex that binds to the tRNA. Okay, and here the hydrolysis of the GTP is very important, where the GDP form. So due to the hydrolysis of the GDP formation is seen, and that helps to the binding of the amino acid tRNA to the A site. Okay, its important role is that. Next point is that EFTU, this is released, leaving the new tRNA is in A site. Okay, in A site. Okay, so what is saying that when the binding is totally completed, listen again, when the binding of the tRNA is completed, then the role of EFTU that becomes stopped or is finished. So what is the function? Is the function of the EFTU now not needed? So that's why it is released, leaving the tRNA. Okay, leaving the tRNA. And here another elongation factor, that is the EFTS. It mediates the release of EFTU. That means EFTU cannot alone, or it's in a single way, it cannot be released from the tRNA. So which can help? This EFTase that helps in the releasing of the EFTU. Okay. Now GDP from the ribosome and the regeneration of EFTU GDP. So again what is happening, that EFTU just released, and so after releasing EFTU, so what happening, again regeneration of EFTU GDP is seen. Clear? So here this is the another important thing is that not included in this translation process. It is one kind of factor or subunit, you can say that, or antibiotic. It is known as the antibiotic. Its function is that this is the chiromycin, and its function is that it inhibits the function of EFTU. Okay, chiromycin is an antibiotic and it inhibits the function of the EFTU. Okay, so this is the process of the decoding. So this is the process decoding, and that is involved in the elongation process. Second is the transpeptidation. What is the transpeptidation? The peptidyl group on P site of tRNA is transferred to the amino acid group in A site. This is the A site, okay? This is the A site and this is the P site. So what is happening here? The peptidyl group that present on the P site of tRNA, this is transferred to amino acyl group in A site. Amino acyl group that is present on the A site and the peptidyl group that is on the P site. So transfer of the tRNA is occurring where the P site of tRNA is transferred to the A site. Okay, transferred to the A site, and through the formation of peptide bond. Here the peptide bond is formed. Here the peptide bond is formed. So processed is what? First, the carboxyl end of the polypeptide chain is uncoupled from the tRNA molecule in the P site and joined by peptide bond to the amino acid linked to the tRNA molecule in the A site. So what is saying that which is present on which site? First you have to understand. The carboxyl end of the polypeptide chain that is uncoupled from the tRNA molecule, and this carboxyl end group or carboxyl group that is present on the P site. So what is happening here? It is uncoupled in the P site and then joined by the peptide bond with the amino acid that is present in the A site. Okay, sorry. Sorry for the disturbances, and please adjust the sound. Okay. So what is saying that carboxyl group is present in the the P site, and this is joined to the amino acid that present in the A site, and by which bond? By the peptide bond. By the peptide bond. And this reaction is catalyzed by one enzyme. This enzyme is known as the peptide transferase. And amino group of amino acyl tRNA, this attacks the carboxyl group of ester and linkage of peptidyl tRNA to form a transhedral intermediate. So what is saying that due to the peptide bond, the carboxyl group and the amino group that join to each other, okay, and due to this joining, what is formed? One intermediate substance that is called the transhedral, it is formed. Okay. Next, this collapses to form the peptide bond and releases the deacylated tRNA. And after this, what happening? The peptide bond becomes collapse. This collapses to form the This collapses to form the peptide bond and releases the deacylated tRNA. Okay. So which are already the substances between which the peptide bond is formed, they can remain, but without formation of the peptide bond, the substances that are present, that means deacylated tRNA, these are released. Clear? And peptide bond formation involves an acyl group O to N. This acyl group is nothing but O and N between the linkage between the O and N. And this migration is occurring, and here the converts an ester to amide. That means here the conversion of ester to amide group. Okay, ester group to amide group conversion is seen. That means peptide bond formation involves what? It involves an acyl group. Acyl group is that the acyl group is that the bond formation between the O and N, and here the transference, you can say, or the not transference, converts means here the ester group that converts into the amide group. Okay. The bacteria, in case of bacteria, peptide transference actively can be detected in 50S subunit ribosomal proteins and involved for this activity. So this is very simple, that in case of bacteria, this peptide transference actively can be detected by which one? This 50S subunit ribosome. Okay, another ribosomal proteins also involved in this situation. So last step is that transpeptidation is the second step, and the last step is that is the translocation. Okay, and so first one the decoding, second one is the transpeptidation, and the third one is the translocation. So including by the three steps, this elongation process is completed. Okay, so last step I have to explain. Okay, so the last part, this is the translocation. What is the translocation? A site and P site tRNAs are transferred to the P site. So what is saying that A site and P site tRNAs are transferred to the P site and E site. Okay. Next point is that, so this is called the actual translocation. So how this is occurring? Here we have written. Here I have written what? The new peptidyl tRNA in A site is translocated to the P site. So new peptidyl tRNA in the A site is translocated to the P site. So here this is a rough diagram how this elongation process is occurring. So what is saying that the tRNA that is present in the A site, it is transferred to the P site. Second point is what? The ribosome, as the ribosome moves exactly 3 nucleotides along the mRNA molecule going in 5' to 3' direction. Simple way, this is occurring 5' to 3' direction, in the 5' to 3' direction. And ribosomes also move in that way. Next point is that this translocation is mediated by the EFG and is driven by the splitting a GDP to GDP. That means hydrolysis of GDP is also present, because without getting the energy, this translocation is not possible. And another elongation factor, that is the EFG, this is also responsible for this translocation. And this hydrolysis of GDP also releases the unchanged tRNA from P site and transfers to the newly formed peptidyl tRNA from A to P. So another important thing is what? The unchanged tRNA from P site, it is also transferred, and the newly formed tRNA, that is also transferred from A to P. So in the this is the point where it is saying that the A site, in the A site that is present the tRNA, that is translocated to the P site. But here what is saying that this the peptidyl tRNA from A to P site. Okay. And this is the same also, but here one thing is written that the hydrolysis of the GTP, that is important here, because hydrolysis of GTP is what? That is the GTP that transfers to formation of the GDP and the inorganic phosphorus. So this hydrolysis of GTP that releases the unchanged tRNA from P site, and in that way this changing is occurred, and this unchanged tRNA moves to the E site. So if you see this diagram, first of all you can see this is the P site and this is the A site, okay, and this is the tRNA, and this adding with the F-mate, okay, F-mate is there, and codon and anticodon I have already told you that if this is codon, that is AUG, then the anticodon is the UAC, okay. So this UAC, this anticodon, and the codon AUG both have to comparatively matched with each other. That means if AUG is there, if here the U that bond only with the A, and another thing is that G that can bond with the C. So these are codon and anticodon formation. If they cannot match with each other, so this bond formation is not possible, okay. In the next step, you can see that the amino acid tRNA and EFTU GTP both enter with this large subunit, okay. So in the P site, you can see that F-mate is already bound here, and another tRNA with the T group that is also bound to the A site, and so that means entry of amino acid tRNA into the acceptor site is occurring. So here this is already bound, and here this now just this is bound with the A site. And in the third step, you can see that peptide bond formation is occurring, and here you can see that F-mate that is already bound to the P site tRNA that form the peptide bond with the T and is bound with the T, okay. That means it is released from tRNA and bound with the T. Next one, you can see that what happened to this tRNA. Now EFG GTP is added here, and here this tRNA that is already bound to the P site, that is released. So what happened to this one that is already formed by the peptide bond and is already present to the A site? What happened to this one? This is translocated to the P site. You can understand now? This is translocated to the P site, and you can see the bond is, peptide bond is already occurred between the F-mate and the T, and the A site now is completely blank. So here another tRNA whose anticodon is matched with this codon, they can now bind here. Clear? So this is the translocation. That means you can see first P site, it is already bound with the initiation codon AUG, and when it is already bound to the P site, now another tRNA with the anticodon that is matched with it, that is bound to the A site, and you can see here both are bound to the P site and A site. But in this stage you can see that P site, this released is its group, and this group is formed, this group just formed the peptide bond with the another tRNA that is present A site. This the group that is present with this A site tRNA, the F-mate form the peptide bond with that group. Clear? And in the last step you can see that formation, the peptide bond already formed to the A site, that is translocated to the P site. So what happened to this unchanged tRNA? This is released, and this is released by EFG GTP. Okay. So hydrolysis of the GDP is also necessary for this. And here another site is also present, that is the exit site, that is the E site. So it transferred to the E site and then released from the ribosomal subunit. Clear? So this is the elongation process I have already explained here. So only one process in the translation, that is the termination process, I have to explain. So it is not possible to explain in this video. So you just have to wait for the last portion or last video where I will try to explain about the termination. If this video is liked by you, obviously you can share with your friends.